Hello, everyone, and thank you for joining us for the 2024 Virtual Water Ambassador Program. The Water Ambassador Program is a partner program between Martin County and the University of Florida IFAS Extension, and it's a program meant to educate residents in Florida about stormwater, water quality, and ways to improve water quality in the area. So today is one of three presentations that we have left for the 2024 season. We have two coming up in September, which is different from what we normally have. We're excited to say that our September 3rd presentation is actually going to be slightly different than our traditional model. We're actually going to be screening a quick video, Emerald Sanctuary, about the health of seagrasses in the state. And then we're going to be having an expert panel talking all about seagrass restoration in the state, including in the Indian River Lagoon region. So this is your opportunity to talk to research and restoration experts and ask them your questions and pick their brain about what the state of seagrasses are in Florida and the Indian River Lagoon. And then our last presentation is going to be on September 17th. This presentation is going to be given by Dr. Mary Lusk, talking about potential nutrient impacts from irrigation overspray. So essentially what happens when your irrigation sprays onto streets and sidewalks, and what's the impact to that to our stormwater. We are also excited to announce our second field trip. So thank you to those of you who were able to join us for our field trip earlier this week to the Ripple Project. It was a fantastic experience, and we received a lot of great feedback. So we're excited for our next field trip. This one is actually tied in to the September 3rd webinar talking about seagrass restoration in the Indian River Lagoon. And we're actually going to be getting a tour of some of the seagrass restoration sites and learn about the research that's happening on site at the Florida Oceanographic Society by one of the producers of the Emerald Sanctuary movie. So you can use the QR code there to register. We will also put the registration link in the chat box. This field trip is limited to 15 people, though, so please only register if you know that you're able to participate. And if you register and are suddenly unable to participate, please let us know so that we can ensure that we allow for the maximum number of participants. We will be recording this, as we do with all of our presentations, and we'll be posting it on the IFAS Extension Martin County YouTube page. So best just to Google that and search for it. And this and all of our playlists are up there from this, the 2024 webinar series, as well as previous years. Please type any questions you have in the chat function. We'll be saving all of our question and answer to the end of the presentation. And with that, I'm really excited to introduce our co-presenters for today's presentation on the long-term effects of exposure to harmful algal blooms, or the LEE-HABs study. Today we have Dr. Rebecca Koszalinski and Dr. Malcolm McFarland. Becca is currently at the University of Central Florida. She leads the Florida Department of Health funded study with a large team of investigators, including epidemiology, water, air, and environmental scientists, bench and nurse scientists, and nursing students to investigate the health outcomes of these harmful algal blooms. Becca has several professional designations. We actually couldn't fit all of the letters next to her name, so I'm not going to try and say them all here, but suffice it to say that she is definitely an expert, and those include Fellow of the Association of Rehabilitation Nurses and a founding member of the Society of Nurses, Scientists, Innovators, and Leaders. Prior to joining UCF, Becca was an associate professor at Florida Atlantic University, Christine E. Lynn College of Nursing. Dr. Malcolm McFarland has extensive expertise in the ecology of microscopic algae. He's acquired over more than 25 years of conducting lab and field-based research, and he's actually been a prior water ambassador webinar presenter on some of these algal blooms and his expertise in the Indian River Lagoon and St. Lucie estuary region. His work has focused on quantitative assessment of algal species abundance and distribution using innovative bio-optical and cell imaging-based approaches. His goal has been to better understand bloom dynamics and the role of algal species in diverse aquatic ecosystems. So with that, I want to thank both Becca and Malcolm for being presenters today. I'm going to stop sharing my screen, and I'm going to turn it over to you. Thank you.
All right, I guess I'm gonna get things started here. So I will share my screen. All right, is everyone seeing that? Yes?
Yeah, looks great. Thank you.
Okay, great. Excellent. Well, thank you very much for having me. I appreciate the opportunity. I'm Malcolm McFarland. I am co-PI on this project we'll be talking about with Rebecca Koszalinski, and my role in this research is as an algal expert, an expert on toxic algae and algal blooms, and how they can be used to look at the impacts on how we can look at these blooms to assess impacts on human populations. So just so we're all on the same page, I thought I'd start off with just sort of a general introduction to the algae that we're talking about, why it's important, and then Rebecca will talk about more specifics about the individual study and what we're doing with human participants to understand more about impacts of these blooms. So my expertise is really in studying these microscopic algae, and these are some examples that you're seeing on your screen of different ones that we have in Florida. These are naturally occurring organisms. They're part of the natural environment. There are various types of algae, but the ones we're concerned with here are these single-celled microscopic algae. They are photosynthetic, so they're primarily plant-like, and that's the role they have in the natural environment. And all the ones we're concerned about generally for this study and for most harmful algal bloom studies are what are classified as phytoplankton. So that means they live suspended in the water, and they sort of just drift around with the currents. They're these passive cells that float around in the water. And most of them are beneficial. Most of these types of algae are critical to aquatic ecosystems, but many of them can grow excessively and sort of grow out of control. And in such cases, they can become harmful in various ways. And in particular, some of them produce toxins that can cause illness and disease in humans and in animals. And as you can imagine, when these organisms grow out of control, they can become a serious problem. So, as I mentioned, there are many different types of these organisms, these algae, microscopic algae. And what you're seeing here is some examples of different kinds when they bloom extensively. When we see a large growth of these single-celled organisms and we accumulate a large biomass, we call that a bloom. And depending on the species, these blooms can look very different in the environment. So what you see here is a whole different range of colors from these blooming single-celled algae. Most of these pictures are right from Florida waters, but you can see things that are bright green, bright orange, brown, or even sort of fluorescent yellow. And this is because they all have different photosynthetic pigments that they use to harvest sunlight and to grow. But these are examples where these organisms have grown to an extent where they can start to cause harm to people if they are toxic, and various problems with the environment. So what kind of problems are we talking about? So, well, if some of these organisms produce toxins, they can be really a direct threat to humans and animals. A lot of people are probably familiar with the Florida red tide, which happens over on the Gulf Coast of Florida most frequently. Every now and then it makes its way over to the East Coast, but pretty much it's usually almost every year on the Gulf Coast of Florida. This is a type of dinoflagellate called Carenia brevis, and it produces a toxin called brevetoxin. This particular toxin is extremely toxic to fish, and so it causes many fish kills along the Gulf Coast of Florida. It's also a direct threat to humans as well. It's irritant, and it can cause irritation to your mucous membranes, your nasal passages, your lungs, and people who have compromised airways, people who are asthmatic, for example, can really be at risk from some of these blooms. Other ways that these organisms can be harmful include things like anoxia in the environment. So when you have large blooms of these algae, as they die, they are consumed by bacteria. These bacteria then consume all the oxygen in the water. And anoxic water can be a real problem for fish. Fish need to breathe as well. If there's no oxygen in the water, they can die. And these anoxic events can lead to massive fish kills. You're seeing in the upper right here an example of a fish kill caused by an algal bloom a number of years ago in the northern part of the Indian River Lagoon. They can also absorb sunlight that might otherwise be available to submerged aquatic vegetation like seagrass. So one of the big reasons we have had a lot of loss of seagrass in the Indian River Lagoon in particular is because of these microscopic algae absorbing all the sunlight and shading the seagrass, not giving it enough sunlight to grow. This has happened in particular due to organism, a type of microscopic algae called Oreoumbra lagunensis. This has led to a lot of very turbid water in parts of the Indian River Lagoon where it's very hard for seagrass to grow. And then, of course, when we have large-scale loss of seagrass, we have other effects like not enough food and starving manatees. Some of these organisms, like you see in the bottom right here, have siliceous cell walls. They're essentially made of glass, and they produce a lot of spines. And even their spines can have spines like thorns on a rose bush. And these can be very irritating to the gills of fish. Particularly, this is a problem in aquaculture in many places. And irritation to the gills can cause fish to suffocate and die. In other cases, some of these organisms can secrete mucus or these mucopolysaccharide compounds that can do things like destroy the weatherproofing on birds' feathers and cause them to become saturated with water and hypothermic. And another way these organisms can be harmful is through economics. So you can imagine large fish kills and discolored water is not really good for local businesses that might make their living based on utilizing these aquatic natural resources that we have in Florida that lots of tourists like to come and see. So why does this really matter? Why are we looking at this? Well, obviously, algal toxins are a direct threat to human health, and we don't really yet have a really good handle on some of the long-term effects of exposure to these toxins. These headlines you see here are some that I've just grabbed over the past few years relating to harmful algal blooms in Florida, and it's definitely something that has caught the attention of a lot of people in the general public. because it is often very obvious when it's happening, and it's of concern. People recognize that this is not a good thing, that we should probably be doing things to mitigate this and make sure that we understand what these threats are. And they're not just a threat to humans and animal populations, but really they can disrupt entire ecosystems and cause really fundamental change in ways that may not be beneficial or that may not be the greatest for us, in ways that can be detrimental to tourism and business. And we have reason to believe that the incidence and the intensity of some of these blooms is really increasing over time. This is probably related to climate change and warmer temperatures, longer growing seasons, different rainfall patterns that help to fuel these blooms by increasing nutrient inputs to coastal waterways, and also just more people in general over time, and more human-induced modification to our coastal ecosystems and our freshwater ecosystems. All these things are combining to really fuel these blooms and increase their incidence over time. So this is something we want to keep an eye on. We want to understand better how it's changing over time and what the potential impacts of those changes might be. So for this study, so that was all sort of a pretty general introduction to harmful algal blooms. And Florida has really become ground zero for a lot of this stuff. There are frequent algal blooms in Florida of varying types. As I already mentioned, the Florida red tide and the Oreo umbra. But the one we're particularly focused on for this study is an organism called Microcystis. And Microcystis aeruginosa is probably the most common species of this. Species in some of these things can be hard to identify. So there may be other species involved as well. But Microcystis aeruginosa is a fairly common freshwater cyanobacteria. So this is actually a prokaryotic organism. And it blooms pretty extensively in almost any freshwater body in the state could be subject to these blooms. It's not just a problem in Florida, it's also a problem in other parts of the world, even other parts of the US, even up in Lake Erie, far to the north of here, they have extensive problems with blooms of this organism. So this is a particularly problematic, harmful species that we have here. What you see on the far left is a colony of many, many individual cells, and they form these sort of big amorphous colonies that you can actually see with your naked eye if you go looking for them. So the individual cells are just these little microscopic green dots that you see all stuck together here. And then at the top, you see an individual cell, one of these cells blown up really big through a transmission electron microscope. And if we look inside these cells, you can see a lot of these honeycomb-like features within the cell. And those are actually gas vesicles, little tiny air pockets within each individual cell. And that makes these cells and the colonies very buoyant. So this particular type of alga really floats very well to the surface and forms these thick aggregations or scums at the surface, which does two things. It makes it very obvious that there's a bloom, pretty easy to detect, but it also has the effect of concentrating all these cells at the surface. And for this one, which produces a toxin that we call microcystin, which you see down here at the bottom, this means that toxin will be very, very concentrated at the surface of the water. And when the wind blows across the surface of the water, it can concentrate even more into coves or along the shoreline. And so you can get patches of very, very concentrated toxin near the surface and near where people might be along the shoreline, swimming or fishing. So this toxin produced by Microcystis is known to be a liver toxin, a hepatotoxin. There are actually more than about 250 types of this particular toxin. The most common one is what you're seeing here. This is microcystin LR, but there are many different variants. Microcystin LR is the one that is produced in the most abundance by these cells. It is also the one that is probably the most toxic. So if you do ingest this somehow, accidentally or on purpose, the effects of this will not be immediate, but essentially what will happen is you can have liver poisoning. Acute effects can be liver failure, but we sort of have a pretty good handle on that. But what we don't really know is what are the long-term effects of low-level exposure to this. It's reasonable to assume that people near these blooms over many years, if these blooms are a regular occurrence, might be exposed to this compound at low concentrations, but over a long period of time. So here in Florida, this organism blooms pretty much every year on Lake Okeechobee. And on the left here, you're seeing a satellite image of Lake Okeechobee. And this green on the surface of the water, which has sort of been blown by the wind into this net-like pattern, this green material is all these floating microcystis that you just saw in the microscope image on the previous slide. So it's many of these colonies aggregated together and accumulated at the surface. And this is a pretty extensive bloom. You can see that it covers quite a bit of area on the lake. And concentrations of the cells and of the toxins will be very high in some of these patches. And of course, Lake Okeechobee is surrounded by a fairly rural area, but the water from Lake Okeechobee is managed by the Army Corps of Engineers. And there are discharges of this water both to the east through the St. Lucie Canal and to the west through the Caloosahatchee River. And at times, the Army Corps has to release water to maintain a safe lake level. And if the lake happens to have a large bloom on it, this can be a problem for these downstream estuaries, the St. Lucie estuary and the Caloosahatchee River estuary, which are both associated with higher population centers along the coasts of Florida, like Stewart and the Cape Coral region. So on the right, you're seeing an example of one of these discharge events transporting a bloom of this algae into the St. Lucie estuary. And so what normally would have been probably brown water that you would normally see in the St. Lucie estuary has this bright green scum on the top of it that is just thick aggregations of algal cells. And toxins in these surface scums can be very, very high. So there is a risk. Probably most people would have the common sense not to jump in this water if it looked like this and try to drink it. But if you are recreating on the water, you can have accidental ingestion, and people's pets are generally not so smart. And there have been a number of incidences where dogs in particular have drunk this water without their owners' knowledge, and that has resulted in the death of the dog. So with respect to people, back in 2018, there was a big event released from Lake Okeechobee that transported a lot of this algae into the St. Lucie estuary. What you're seeing here on the left is measurements of the microcystin toxin in water samples taken from a time period from around June to September in 2018. The circle size here indicates the concentration of toxins in that water sample. And some of these water samples have concentrations very low that would normally not be of concern, like 0.01 parts per billion, but many of them had greater than 200 parts per billion, which is very high toxin concentrations. The World Health Organization recommends an upper limit of about 8 parts per billion for recreational use of waterways that might contain these toxins. And so what we were able to do, this was with our colleague who is part of this project, Adam Schaefer. I was accumulating toxin measurements from water samples, and he was measuring microcystin concentrations in nasal mucosa from samples taken from people who live and work around the St. Lucie estuary. And what we saw was at the when we were first able to get samples from people, we saw initially rather high concentrations, well, relatively speaking. Actually, high is a relative term in this case. We don't know if these are concentrations of concern, but we were able to measure them in people's nasal swabs. We saw initially relatively high concentrations that decreased over time. And this decrease over time corresponded to the decrease in the toxins in the water samples. So it seems there is a correlation here between the exposure that people experience to the toxin and the toxin in the environment. So that led us to question, what is the significance of this? Is this, apparently people are being exposed, but is this a problem? Are these toxin concentrations enough that we need to worry about the potential health effects? So that was sort of the idea behind this further study, this longer term study, where we would be able to look at people over time, over a longer time frame, and see if we see health effects, not only toxins in the environment and potentially toxins in people, but do we see health impacts from that apparent correlation? So since that time, we have been, or I have been, as part of my involvement in this project, have been monitoring these algae in the environment. I do that with a variety of methods. We go out, we collect water samples. We also make use of the extensive water samples that are collected by South Florida Water Management District and Florida DEP. They are measuring for these toxins as well, so we make use of that in addition to samples that I might be collecting myself. We have some interesting autonomous instrumentation that we have installed on Lake Okeechobee to monitor blooms. This is an example of an instrument called a sea prism, which measures the water color. And so if we see a lot of green water, we know there's a lot of toxic algae present. And then we also use satellite imagery to monitor these blooms. Since the microcystis in particular floats really nice at the surface, it shows up pretty nice in satellite imagery when it's abundant enough, and we can use that, especially on Lake Okeechobee, since it's a large water body. We can use that to track blooms and determine when it's very abundant and when there might be risk to human populations. So we have started to assemble this long-term data set of samples and toxin concentrations, and from my contribution to the project, this is building this database over time of environmental samples. And we're really focused on sort of 3 areas here. So our initial studies were in the Stewart area on the east coast of Florida, but we are monitoring algal and bloom activity in Lake Okeechobee, which is where a lot of these blooms start, and we see very regular bloom activity every year. And then also over in the Cape Coral area. So both of these are downstream regions from Lake Okeechobee where the microcystis and the toxins can be transported to the east through the St. Lucie Canal or to the west through the Caloosahatchee River towards these regions where we might see health impacts from people exposed. So that's a summary of my contribution to this project, and I guess a bit of an overview of HABs in general and this particular type of HAB that we're studying. And I guess now I'll turn it over to Rebecca. Unless there's any questions for me right now, maybe we should just keep things moving.
Are you ready? So thank you so much, Malcolm, for that. That was fantastic. So you may be wondering how a nurse came into this project. How did the nurse become PI of this particular wonderful project? So before I was a nurse, I was actually in tissues. I was in pathology and histology. So I came out of that bench science world, which I love and obviously missed because here I am. So working in tissues then was a wonderful bridge to start working with this team. I came into this a year ago as PI at FAU. I've since transitioned to University of Central Florida, where we are continuing this work. And Malcolm and I have been tasked with leading this wonderful group and this science. So as Malcolm said, we don't know the long-term effects. That's really the question here. There's quite a bit of data about what it means to swim or ingest or have that direct contact with harmful algal blooms, but we don't know about these long-term effects. So this is the longitudinal study that we've been working on, knowing that, again, we have this data, we have basic understanding of the health outcomes, but there's so much more to learn. So the aim of our study is to evaluate the short and long-term health effects of exposure to HABs, specifically right now in the state of Florida, and we're using a collaborative multi-site approach. I think you saw on the first slide, we have Florida Gulf involved, we have Harbor Branch, we have FAU, the Clinical Research Unit, which is housed in the College of Medicine, and now UCF. So this is a large group, many pieces, many working parts, and we're all just trying to answer this question. So again, I'm not sure how much of this I want to repeat, but what we're looking at, I'm going down to that last bullet point, is about the human exposure to bloom toxins, and we're looking at this in biological samples obtained at the time of the interview. So this is what we're doing that is unique, not to mention the health outcomes piece of it, and looking specifically at participants who live in those areas that Malcolm so wonderfully, developed for us to be able to see that map. So we're looking at toxins and swabs of the nasal mucosa. This is not like COVID. They do it themselves. It's a small Q-tip. They're not doing the full extension into that, those areas that are uncomfortable. They're leaving us some urine, and we're taking a blood sample. Now, the nasal swabs and the urine and some of the blood go off to a lab for testing. We also use green water, which looks at whether or not we have any toxins that are apparent. And we're looking at whether or not, over time, now finished 5 years, do we have anything? Are there any outcomes that could suggest there are negative health outcomes related to exposure to these HABs? Can you go to the next slide, please, Malcolm? Thank you. So I just said this. Oh, right. And I did not say Colorado State. We have John Reif also working with us. And Malcolm did mention Adam, who really, between Adam and Malcolm, really spearheaded this and sent us off into this area of research. You want to go to the next one, please? This is something else that we're doing that we are quite pleased with. We not only have these major investigators, we're also including students. We had an undergraduate student, Antonio Roll, who spent time in our clinical research unit, learning not only how to work at the bench, but also how to do sampling out in the community. So he'll be able to use that research as he goes forward. And then we have a PhD student who's Judita Kocholek, and she's also the head of the CRU. So we're eager to include students. You might want to tuck that away, perhaps, if you think of someone who might want to get involved. We're happy to talk with them. This is also a nurse-led interprofessional team. There is another team that we are aware of that is doing this, having a nurse involved in the health outcomes. And we've published 3 papers about the HABs problem in Florida. We are just finishing up, as I said, year 5, so we are in data analysis. And of course, we will continue forward with additional funding in the next year. You go ahead, Malcolm. Thanks. So you've already seen the areas, which is great. So Stewart, St. Lucie, Fort Myers, Cape Coral, Okeechobee, and Martin. I saw a question in the chat. If you don't mind, I'll just jump to that. How do you get involved? Essentially, if you're an adult and you come to these collection times and places, you can enroll as a participant if you would like. We do an intake health survey. It's very brief, at least we think so. We do that blood draw. We ask you to do a nasal swab again on your own and to leave a little bit of urine for us. Next slide, please, Malcolm. These are the 3 papers that we have published at this point about this particular stage. And I'll get to that question in just a moment, Vincent, great question. So we first used, oh, I just lost the screen. A unique testing process, and that was led by Brady Cunningham, who's at the CDC, to help guide how we would do this. Since then, we have done publication by Adam Schaefer, who was the first author on that, looking at exposure to these microsystems. And then the most recent paper is John Reif, looking at the frequency of symptoms compared to the exposure of cyanobacteria blooms in Florida. And you might have questions about that, but we'll go on and we can always answer the questions at the end. So these are the identified gaps. There's a lack of comparative data collected during HABs versus non-HABs. As Malcolm was saying, you can see the HABs, you can see when there is a bloom, but we don't know what the outcomes could be related to that residual, if you will, the non-HABs period of time. So we don't really know what that means yet. And exploration of design possibilities that will better accommodate long-term exposure studies rather than just these just cohort. We are trying to, explore designs that will better represent what we're trying to do in this longitudinal study, which is a challenge. Go ahead, Malcolm. And these are our major takeaways at this time, on this day. At 12:30, these are our major takeaways because we know it's always changing. We have a biorepository, very important to us. We're going to begin secondary data analysis to see exactly what we have in that biorepository over 2 years. And that includes also the urine samples, also bloom versus non-bloom. As I just mentioned, we're looking at whether or not there are differences or, what is it that we actually have in the data. Again, our long-term cohort challenges, recruiting new participants each year because we do have loss each year. People do move out of the state. We do have snowbirds that may decide not to come back down the next year. And so that's always a challenge. And possibly reduce community interest during non-bloom time periods. Again, if you can see the bloom, then you know that this is going on. If you don't see the bloom, people may be a little less interested because it looks like there's no issue. So that can also be a challenge for us. And then we all know about the unpredictability of HABs blooms. So you may go to bed one night and there's nothing on the lake, but you get up the next morning and there might be evidence of blooms. And so we're always very keenly interested to know if there is something happening on the water so we can get out to it and collect our samples. All right, next slide, please, Malcolm. We need to acknowledge our funders, Florida Department of Health, and our community partners on this study, Volunteers in Medicine in Stewart, Cape Coral Department of Public Works, and the Okeechobee County Health Department. If you know any of these people, say thank you. We're very pleased to have them on board with us. We have the FAU Division of Research and the Clinical Research Unit, and of course now we're adding the UCF resources. We're also thrilled to have Green Water Laboratories on board with us. They're testing for the bacteria and the cyanotoxins, very, very important. I think that's probably the last slide. That is the last slide. All right. We'd love to hear your questions.
Should I keep sharing the screen?
No, I think you can take it down with just the big green question mark. I'm trying to find something that would look like algae. I
Yeah.
did see, go ahead.
We have a bunch of questions in the chat and I can go through them it's in the order. And then if you would be willing to also, you would sort of mentioned the paper that was published that talked about the symptoms. And if you can touch on that a little bit more, but let me go through the questions in the chat first.
Sure.
So we have a question about the actual toxins and whether or not you have to be in the water to be affected, or if you can be affected through airborne aerosolization of the toxins.
Go first, Malcolm, and then you can follow up. So
Sure.
the answer is yes, it can be in the air, and we do have evidence of that. The most typical route, of course, is being in the water. We're thinking also about fisher enthusiasts and kayak, people who are out on the kayaks and boating and such, and people who might actually be putting their hands in the water, anchors and such and lines and whatever they're doing might actually have that physical contact. But yes, it can be airborne.
Okay,
Yeah,
great.
I think the airborne issue is one of the big unknowns, and that's something we're trying to address. So Mike Parsons is part of our study, and he is really focusing on taking those measurements that will tell us, or at least hopefully tell us, how much of these toxins are actually in the air during a bloom event. And so that'll really help us understand better the degree to which people might be exposed.
Great, thank
Right.
you.
And we don't want to, create too much concern. It's not that, necessarily if you're near the bloom that you are getting a full effect. It's just that it can be. And as Malcolm said, yeah, Mike Parsons is going to help us sort that out as well.
So we have someone who is showing some interest in becoming a participant. So I think you addressed the first part of their question, which is whether or not the study is closed to new participants. And if not, what is the criteria to participate?
Being an adult, being able to travel to the collection site. And anyone can get involved. We're especially looking for people who are recreating on the water where there could be blooms. That's especially important to us, but we are not excluding people who are not.
And then, so Vincent asked if there are any upcoming collection events, or if they're interested in becoming a participant, how should they get in touch? What should they do?
We do have a website, or no, sorry, we will have a website. We do have a dedicated email, and I will send that out to you, and maybe you could disseminate that.
Yeah, we can put it in the chat right now. If one of you have the ability to enter it in the chat, that would be great. But we can also send it out.
Sure, I just have to go look, unless you have it, Malcolm.
I don't have it off the top of my head.
Okay.
No, sorry. But at this point, we don't have any collection events scheduled right now, right? I think, but we're looking at some point in the fall, as we get into the fall.
We are between years right now. And so we're planning for the next phase. We're hoping to get back out by October. That's our goal. And we'll make sure that a press release goes out, of course, at that time. Because we don't know where it's going to be yet. We're not quite sure where we're going to start. Which one of those data collection sites. So I'm sorry, that's still up in the air.
That's okay, thank you. The next question comes from Michelle. She says that we are finding microcystins in community stormwater ponds, and some of these ponds are used for irrigation. DEP doesn't sample these because they're not public waters, and the only requirement is that the pond has a sign posted, but it can still be used to irrigate homeowner properties within the community. Do you know of any studies in these areas? Since the water is used for irrigation and it aerosolizes, I'm wondering if anyone is looking at the health effects of these practices.
That's a great question, and I think it's not something that we probably have been able to capture in our study so far anyway. I think a lot of the issue is it logistically just becomes very difficult to track down some of these very smaller localized blooms and associate people with those. It would be great to have people who are aware that this might be happening adjacent to where they live, or if they're using this water for irrigation, if they could help join the study. But we've had to focus, because we're sort of covering a pretty large geographic area, we've sort of focused over broader scales than that for this study. But yeah, like I said, the microcystis can pop up pretty much in any freshwater body, you do want to be. Thankfully, it's pretty obvious when it's there because the water will turn green. But it would be great to include people who maybe have realized that this is something that's in the environment near them to participate in the study. That would be great. But it's really hard for us to track down these little patchy in blooms in the environment ourselves.
So I'll put a plug in for Michelle. She's one of our colleagues at UF-IFAS over Manatee County. So if you're looking for a collaborator over on the West Coast, she's fantastic.
Oh, cool. Good to know.
I think one of our participants actually posted contact information and an email. So hopefully that's the correct information. Thank you, Maya, for posting that in the chat. Kay asks, how does this study compare to Dr. Backer's CDC, the Moat Marine Lab study in 2006-2007 in Michigan and California on recreational exposure and microcystin?
Well, first of all, we're not, and I'm not really familiar with this study to the extent probably that Malcolm is, but we are not looking specifically at recreational. We do have a larger population of older adults, especially the snowbirds, but people who have lived on that lake or on the lakes for generations. And we're interested in that over a long period of time.
Yeah, I think I'm not sure I remember the details of that study, but I think the difference here is the scale that we're looking over a large geographic area, and the time scales that we're looking at over a long period of time, and trying to get at this low-level exposure through aerosolization.
Great question. Thank you.
Yeah, thank you. We don't have any other questions in the chat right now. Wait a few more minutes, see if any new ones come in. In the meantime, would one of you be willing to elaborate a little bit more on the symptoms of microcystin exposure from the paper, the Schaefer et al. Paper?
I'll start, Malcolm, and then you can follow up again. It seems to work well, right?
Well, you're the person expert, so
the human subjects and symptoms, and yes. So we do see respiratory symptoms. That seems to be the most common. We do see some derm symptoms if they've actually been in the water, rashes and such. We only see GI symptoms if they have ingested that water. Otherwise, or as Malcolm said, getting a little bit in your mouth while you're swimming. And if there's a large bloom and they're swimming in it, well, then they might accidentally swallow it. Generally at this point, we're just seeing GI upset, typical flu-type symptoms, not terribly serious. It's only when they are ingesting it in larger amounts that we might see that hepatic failure. And that is the most dire that it would be for the symptoms is leading to hepatic failure. So we do see the symptoms, and the next paper, we are now going to sort this out further. drill down and see what we really do have and compare it to when we do have the blooms. And that's another question, seeing the symptoms. Again, is this a bloom season or a non-bloom period? And what are the symptoms? What are the levels of symptoms? I think Malcolm mentioned that, we may see an elevation, but we don't really know what that means yet. We don't have the metrics yet to say, if this, then this is what we expect will happen. And again, that's what we're trying to learn.
Thank you. Malcolm, did you want to add anything to that?
Oh, I think that sums it up pretty good. The low level exposure, the symptoms are probably a little more, they're not going to be as dramatic. They're not going to be the same as an acute exposure. So I think we're looking to try and tease out something that is a little more subtle from the data. So it's a little trickier. You need larger data sets and you need longer time frames to be able to look at that. But then there's also the issue of, are there increased incidences of things like liver cancer associated with these populations who might be exposed over a long time? And that's not something you're going to capture probably with over a short time scale. You really need more broader scale data to capture something like that.
Thank you. And Rebecca, I saw that you acknowledged the last comment in the chat about the interested participant, someone who's a diver and spends a lot of time in the water and is really interested in participating in this study. So I want to thank you both for your willingness to present to the water ambassadors, and hopefully you'll get some new participants as part of your study out of this presentation as well. And with that, I want to thank you all for participating in our webinar series, and we hope to see you during our next webinar on September 3rd. So thank you again, everyone.
All right. Thank you very much,
Thank you,
everyone.
Thanks.